Hello, my dear students. Welcome you all to Sri Jain Rank Zone classes. I'm your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, in the last lecture, we have started enzymes. We have completed the mode of enzyme action, the law and the key hypothesis, and the induced fit hypothesis. We learned about the different factors which affect the catalysis of a particular bioreaction or biological reaction with the help of an enzyme. What are the different factors like pH, concentration, temperature, radiation, okay, protein, poisons, okay. Now we are going to start with our favorite Neet time, and then we will further talk about both the mechanisms in detailed version, that is lock and key hypothesis and induced fit hypothesis, and the difference between them. Now, so according to Neet time, both in cells and extracellular fluid, the dibasic phosphate, and monobasic phosphate. The dibasic phosphate, it means it have donated two protons. Okay, so it has donated two protons. It is having a basicity of 2, and monobasic is having basicity of 1, H2PO4 minus. They act as an acid base buffers to maintain the K plus concentration of the extracellular fluid, the Na plus concentration of the extracellular fluid, Na plus concentration of the cellular fluid, KH plus concentration of the cellular fluid. So Na and K, they are basically balanced by the sodium potassium pump and the electrolytic reabsorption, selective reabsorption of kidney tubules. H plus concentration is to be maintained because if the H plus concentration outside the extracellular fluid, if it increases or decreases, it can lead to the conditions like acidosis and alkalosis. Okay, when the body will have the blood pH will be decreased or it will be increased. So these 2 points you have to remember, that the cells, they maintain the H plus ion concentration by having a reflex or by using a buffer of natural buffer of dibasic phosphate and monobasic phosphate. Understood? All the following statements are correct except mitochondria are rich in manganese. Molybdenum is necessary for fixation of nitrogen catalyzed by the enzyme nitrogenase. Magnesium is essential for a large number of enzymes, particularly those utilizing ATP. Calcium and magnesium have no effect on the excitability of nerves and muscles. We have to find out the incorrect statement. Okay, so the incorrect statement, first statement is correct, mitochondria are rich in manganese. Second statement is also correct, molybdenum is necessary for the fixation of nitrogen enzyme catalyzed by enzyme nitrogenase. Okay, and the third is also correct, that magnesium is necessary or it is essential for the large number of enzymes, particularly the hexokinases, the kinases enzymes which are involved in the uh, production of ATP or utilizing ATP. Okay, calcium and magnesium, they have effect on the excitability of nerves and muscles. So D is incorrect. D is incorrect. The hormone adrenaline, that is epinephrine, is formed from which of the following amino acids? Okay, there is one amino acid which is involved in the formation of adrenaline, that is epinephrine, which is a neurotransmitter of sympathetic nervous system, that is tyrosine. Okay, so the hormone adrenaline is basically made up of a following amino acid, that is tyrosine. Tyrosine is involved in the formation of the epinephrine, that is adrenaline. Also, it is involved in the formation of melanin as well. Okay, so please remember that tyrosine, it helps to make melanin, to make these uh, mediators, neurotransmitters, right? It helps to make thyroxine as well. Thyroxine. Okay. And melanin. Now coming to next, skin pigment melanin is formed from tyrosine. Which one of the following is a neutral amino acid? So the neutral amino acids, the acidic amino acids, the basic amino acids. So glycine is a neutral amino acid. Glycine, alanine, valine, leucine, and isoleucine, they are neutral amino acid. Glycine, valine, leucine, isoleucine, they all are Examples of neutral amino acids. Prolamines. Prolamines, they are associated with nucleic acids. They are storage proteins, structural proteins. Okay, C and D both options are, same. So prolamines, they are basically, prolamines along with albumins, globulins, all these proteins, they are storage proteins. They are basically, they act as what? Storage proteins. Like, well, whether we talk about the prolamines, okay, prolamines. So, yeah, it is a storage. Albumins, globulins, they are reservoir storage proteins. Cheese is a cheese is a protein which resembles the myosin. Okay, cheese is a myosin. Lysine is an essential amino acid because it is not formed in the body and it has to be provided through diet. Okay, the essential amino acids, the semi-essential amino acids, and the non-essential amino acids. Please remember the names. It's very important. The essential, non-essential, and semi-essential. Okay. Now we are going to talk about the site of enzyme action. Enzyme basically they help to alter the rate of reaction. Enzymes they help to provide a alternate pathway for a reaction. Okay, they help to provide alternate path to the reaction. They help to provide alternate path to the reaction by decreasing the activation energy. Okay, so the site of enzyme action, this is the main function of enzyme. They help to decrease the activation energy. So previously, when the less fraction of the molecules they are having that, or they are able to reach up to that activation energy, they will be only converted into the product. That is known as what? The probable density, okay, or the collision frequency. So enzymes they work same as you have studied in chemistry about inorganic catalysis. Okay. So the site of enzyme actions are intracellular as well as extracellular. Intracellular enzymes, the endoenzymes, they are dissolved in cytoplasmic matrix. Okay, the 11 enzymes that are necessary to change glucose into lactic acid. Okay, similarly, several enzymes which are bound to the particles such as ribosomes, mitochondria, and chloroplast. So inside cell, whatever the enzyme and enzymatic activity is happening, that is endoenzymes. Extracellular enzymes are secreted by the cells and they work outside the cell. They function outside the cell, like the digestive enzymes. Okay, like salivary amylase, gastric, pepsin, pancreatic lipase, all are extracellular enzymes. Now, in the previous lecture, I made you understand about the induced fit model and lock and key model. So, what are the basic differences between these two? Okay, first of all, you should remember the name, the induced fit hypothesis, and the lock and key mechanism. So, the induced fit hypothesis was proposed by Koshland, and the lock and key was proposed by Emil Fischer. Induced fit model, a model for enzyme substrate interaction in which the active site of the enzyme does not completely fit into the substrate. So according to Koshland, there is no active site as such because according to Koshland, the shape of active site becomes compatible according becomes compatible according to the shape of substrate. Okay, when the moment substrate will attack on the active site, then only the shape of the active site will change. The active site is having two groups, buttressing group and catalyzing site. Buttressing site and catalyzing site. The substrate attached to the buttressing site. Substrate attaches to the buttressing site. The moment it happens, the catalyzed site, the catalyzing site, it will break the bonds. Break the bonds. Okay. So this is the induced fit hypothesis which was proposed by Daniel Koshland. Lock and key. This is the second model for the enzyme substrate interaction, okay, in which the active side of the enzyme completely fits with the substrate. The active side of the enzyme does not completely fit to the substrate. The active side of the enzyme precisely fits into the substrate. The active side of the enzyme has to undergo conformational change in order to improve binding. Okay, that is why I have mentioned change the shape, change in the conformation. It describes the specificity of active site of the enzyme to particular substrate. Mean the conformation will not change. Okay, it describes the specificity of active site to a specific substrate. The active site of the enzyme contains two components, buttressing and catalyzing site. The active site of the enzyme contains only single entity. There is a separate catalytic group in the enzyme. Okay, there is no separate catalytic group in the enzyme. The active site of the enzyme is not static. It is not static. The active site of the enzyme is static. The transition state develops before the reactant undergoes changes. No transition state. The transition state like this. Transition state like enzyme plus substrate leads to the formation of enzyme substrate complex, and then product. Okay. There is no transition state in case of lock and key mechanism, but there is a transition state in case of induced fit model. The catalytic group weakens the substrate bonds either by the nucleophilic or electrophilic attack. Okay. No weakening of the substrate bonds. So the catalytic group helps to weaken the bonds of buttressing and the substrate, okay, by the nucleophilic or the electrophilic attack. Understood? Describes the mechanism of non-action over competitive inhibitors. Okay, describes the specificity of active site of the enzyme to a particular substrate. So this is the difference between the induced fit model and the lock and key mechanism. Diagrams you have already seen. Now we are going to talk about the inhibition of enzyme action. Okay, what are the different types of inhibitors? What are the different mechanisms of the inhibition? Understood? Now, competitive inhibition. Basically, you have to study the three basic types of inhibition. One is competitive, one is non-competitive, and the other one is allosteric. Okay, so the competitive inhibition. The competitive inhibition says what? According to competitive inhibition, the enzyme action may be reduced or inhibited in the presence of a chemical or a substrate or an opponent, analog that is similar to the substrate in the molecular structure known as competitive inhibitor. So there is a substrate and there is a analog of that substrate or opponent of that substrate which will trying to mimic that substrate to attach to the active site. The inhibitor competes with the substrate as both can attach to the enzyme for the substrate binding site. Both can attach to the enzyme at the substrate binding site. Understood? Consequently, the substrate product reaction does not occur. Consequently, the substrate product reaction, it doesn't occur. Okay, it doesn't occur. For example, inhibition of succinic acid or succinic dehydrogenase enzyme by molecule malonate, which is similar to the substrate succinate in structure. It means we are having succinate as a substrate, okay? Succinic dehydrogenase as an enzyme. Suppose this is succinic dehydrogenase. This site is for attachment of This is succinate. If malonate comes as a inhibitor, malonate comes as an inhibitor, this is known as competitive inhibitor. Understood? The moment competitive inhibitor attaches, okay, the moment competitive inhibitor attaches, the product is written, the product is not obtained. The substrate product reaction does not occur. Okay, so this is the inhibition of succinic dehydrogenase by molecule malonate. So this is competitive inhibition. There is no chemical change which happens. Okay, just the enzyme or the active site is occupied. No chemical reaction. Understood? Now, non-competitive inhibition. Okay, non-competitive inhibition. Here you can see the substrate and the opponent or the analog, they are not matching to each other, so they are not in a competition. They are not in a competition means substrate and inhibitor, they are not fighting for the same site, for active site. For example, cyanide kills an animal. Cyanide kills an animal as it inhibits the cytochrome oxidase. Cytochrome oxidase is very much important for ETS, electron transport system. Okay, which is a mitochondrial enzyme required in cellular respiration. The cyanide, it acts as an inhibitor for the cytochrome oxidase, which is a mitochondrial enzyme for cellular respiration. Here the inhibitor cyanide is different from the substrate cytochrome. Substrate is cytochrome, enzyme is cytochrome oxidase, okay, and thus does not bind to the enzyme at the substrate binding site. However, the inhibitor binds at some other site of the enzyme. The substrate binding does not occur, however, the reaction for the product formation does not take place. This is enzyme, this is substrate, this is inhibitor. The inhibitor is attaching on the different side, okay, not to the substrate binding side. It is not binding to the substrate binding side, it is the different side, okay. So no reaction happens. Inhibitor binds to the allosteric site. So these are what, you can say that these are the complete inhibitions, and these inhibitions, they help, or they help the inhibitor. Okay, these process of inhibitions, they help the inhibitor to meet out their motive, okay, to stop the chemical reaction. Understood? As the conformation of the enzyme will change, the shape of the active site will change, and now no other substrate can attach to that site, and that is why there is no reaction. Allosteric modulation or feedback inhibition. Allosteric modulation or feedback inhibition. The activities of some enzymes which form a part of a chain of reactions like metabolic pathway are regulated in a cell. So according to the feedback inhibition or the allosteric modulation, it is like that the activities, there are some enzymes and the activities of those enzymes which are the part of the chain of a reaction like in metabolic pathway are regulated in the cell. Okay, for any biochemical pathway, like whether we talk about glycolysis, Krebs cycle, or electron transport chain, the enzymes which are helpful or which are important for conveying those biochemical pathways. Okay, so now these enzymes and the activities of these enzymes are regulated inside the cell. There are specific low molecular weight chemicals that are the product of another enzyme further or in the chain act as an inhibitor. Now, these enzymes which are involved in that biochemical pathway, okay, these enzymes produce some low molecular weight chemicals, okay, which further act as an inhibitor in the same reaction. Such a modulator substance binds to the specific side of the enzyme, which is different from its substrate binding site. Now, the modulator substance which is going to modulate to change the conformation of the enzyme, it will binds to a specific site of the enzyme, but that specific site is not the substrate binding site. That specific site is known as what? Allosteric site. This binding increases or decreases the enzyme action. Such enzymes are called allosteric enzymes. Okay, means like for example, if I will tell you that this is a home, this is a home. Okay, now this home is basically having two rooms, room number 1 R1 and room number 2 R2. Now room 1 is general a substrate binding site. Substrate binding site. So normally the room 1 is occupied by the roommate or the person. But if the wrong roommate or more than one roommate, more than the required roommate, they are going to attach with this R1 active site or the substrate binding site, then there is a accessory R2. This R2 will help to either stop the reaction or facilitate the reaction. Okay, like for example, someday the food prepared by your mother is suppose it is very delicious and your hunger it increases tenfolds. Now you are just eating without caring and without having a point of conscious in your brain that you might suffer from indigestion, but your mother is having a allosteric modulator, eno, gelusil, digene. So they will help to, increase or they will help to decrease the rate of reaction the moment substrate binding site is already occupied. Okay. So these are allosteric modulations or feedback inhibition. Okay. Understood? So these are like this. Suppose this site is for activator, this site is for substrate, and this is for inhibitor. Okay, if inhibitor binds or attaches to this side, okay, if it binds or attaches to this side, okay, so this inhibitor will change the conformation, it will change the conformation, right? So depending upon these inhibitors there is a feedback which is generated. Okay, whether the inhibition is required or inhibition is not required. Okay, so all these sites other than the substrate binding sites, they are known as what? Allosteric sites. These allosteric sites are known as what? Modulators or the effectors. They are of two types, either activators or limiters. Either they are activators or they are inhibitors. Either they activate or facilitate the process, or if they are inhibitors they will inhibit the process. Understood? Like the enzyme phosphofructokinase is activated by adenosine diphosphate. So adenosine diphosphate is a activator for enzyme phosphofructokinase, and it acts as a inhibitor for ADP. ATP. So ATP is basically the inhibitor for phosphofructokinase, but ADP, adenosine diphosphate, is an activator. So these are the feedback mechanisms which are generated by, you know, during requirement. These are known as what? Allosteric modulation or feedback inhibition. Then isoenzymes or isozymes I have already discussed. Isomers, isomers, okay, like lactase, lactic dehydrogenase, okay, it is having five isoenzymes. Alcoholic dehydrogenase, I mean they have same molecular formula, okay. Now, I mean they are basically prepared isozymes. They are basically prepared from the same parent, okay, gene, okay. This is induced with hypothesis. Now you can see the buttressing group, and this is the catalytic group. the substrate, it will attaches to the buttressing group, and the conformation of the group was changed. You can say you can see the shape of the enzyme is modulating, it is getting changed, it is getting adapted according to the shape of the substrate. Okay. Catalytic group, it will help to break the bonds. Catalytic group, it will break the bonds, okay, between the buttressing group and the substrate. Some examples of inhibition. Hexokinase, it will be magnesium ion catalyzed, which changes glucose to glucose 6 phosphate in glycolysis. Decline in enzyme activity by allosteric effect of product is called feedback inhibition. Allosteric inhibition of hexokinase by glucose 6 phosphate is a kind of feedback inhibition. Now, what exactly they are trying to tell? They are trying to tell that if in blood the glucose concentrations are high or the glucose concentrations are low, okay, then accordingly the feedback inhibition is generated. So hexokinase will change the glucose into glucose 6 phosphate. Decline in the enzyme activity by the allosteric effect of the product is called, okay, feedback inhibition. Mean glucose 6 phosphate is the product and it is the allosteric product. Meaning it will regulate its own catalysis. It will regulate it in its own catalysis. Okay. So the allosteric substrate will attach to the allosteric site again to control the rate of the reaction. Okay. So it will inhibit the activity of hexokinase by glucose 6 phosphate. So this glucose 6 phosphate will attach on the allosteric site. Will attach. I mean, this is the product. The product after its formation, it will attach to the allosteric site to control the further reaction, to control the activity of hexokinase to make more glucose 6 phosphate. So hexokinase will become relaxed. Okay. Enzyme phosphofructokinase is activated by ADP and inhibited by ATP. Another example is inhibition of threonine deaminase by isoleucine. Threonine deaminase is inhibited by isoleucine. Amino acid isoleucine is formed in bacterium E. coli in a 5-step reaction from threonine. When isoleucine accumulates beyond a threshold value, its further production stops. So another example is inhibition of threonine deaminase. Threonine deaminase is inhibited by isoleucine. Amino acid isoleucine is formed in bacterium E. coli in a 5-step reaction from threonine. So threonine makes isoleucine. When isoleucine accumulates beyond a threshold value, it further production stops. They are known as what? The product allosteric feedback inhibition. Product allosteric feedback inhibition. Understood? Succinic dehydrogenase, succinic acid is the substrate, malonic acid is the competitive inhibitor. This is the example I've already told you in the previous slide. Okay. Inhibitors of enzyme activity. Any substance that can diminish the velocity of enzyme catalyzed reaction is called inhibitor. It is known as what? Inhibitor. Okay. Reversible inhibitors bind to enzymes through non-covalent bonds. Reversible inhibitors, they bind to enzymes through non-covalent bonds. Dilution of the enzyme inhibitor complex. Dilution of the enzyme inhibitor complex results in dissociation of the reversibly bound inhibitor and recovery of the enzyme activity. So there are two types of inhibitors, reversible and irreversible. Reversible inhibition, in which the enzyme is attached to the reversible inhibitor by non-covalent bonds. So if the dilution of the enzyme will happen, the bonds will be broken off, they will dissociate, and the inhibitor will become free. Okay, and the enzyme will become free. The enzyme will become free. Irreversible inhibition occurs when the inhibited enzyme does not regain its activity. When enzyme is not letting free upon dilution of the enzyme inhibitor complex. Some irreversible inhibitors act by forming covalent bonds. They act by forming covalent bonds with a specific group of enzymes. For example, neurotoxic effects on certain insecticides. Certain insecticides, they are being eliminated from the field by the effect of neurotoxins, and these neurotoxins, they basically jam the site where neurotransmitters are released are due to the irreversible binding at a catalytic site of the enzyme acetylcholinesterase. So acetylcholinesterase basically it helps to break the bond. It helps to regain or recover the enzyme acetylcholine. Okay, so it will break the bond between acetylcholine. So if that bond is not broken, then the nerve transmission will not be conducted in the second time. Okay, because free acetylcholine is needed for neurotransmitters for the synaptic effect. Okay, so these are irreversible inhibitions. Okay, the two most common encountered types of inhibitions are competitive and non-competitive. Apart from that, allosteric modulation or feedback modulation is also here. Then protein denaturation, inhibition by protein denaturation is also there. Okay. Feedback or end product in case of formation of isoleucine, you don't have to go through the whole. Just you remember the threonine deaminase, and the isoleucine. Okay. In the product threonine, the product isoleucine, which is formed in the excess amount, will allosterically, inhibit the threonine. Competitive inhibition, I have already told you. Now, what are the kinetics of competitive inhibition? Kinetics of competitive inhibition. This type of inhibition occurs when the inhibitor binds reversibly to the same site that the substrate would normally occupy, therefore competes with the substrate for the site. Effect on Vmax. Vmax is that velocity, that maximum velocity when all the substrates are attached to the enzyme. The effect of competitive inhibition occurs when the inhibitor binds reversibly to the same site that the substrate would normally occupy, therefore competes with the substrate for that site. Okay, so there is an effect on Vmax. Okay, effect on Km. Km is that amount of substrate, that concentration of substrate on which or through which the half of the maximum velocity of a reaction is achieved. The competitive inhibitor increases the apparent Km. Okay, if the apparent Km is increased, mean more and more amount of substrate is required to reach up to the maximum half of the maximum velocity. Okay, it means it is inhibiting or slowing down the reaction. The competitive inhibitor increases the apparent Km for a given substrate. This means that in the presence of competitive inhibitor, more substrate is needed to achieve half Vmax. Okay, half of the maximum velocity. Sulfur drugs for folic acid synthesis in bacteria and inhibition of succinic dehydrogenase by malonate. So sulfur drugs, okay, they basically help to destruct or help to destroy the receptors which are responsible for the folic acid synthesis in bacteria. Okay, and the inhibition of succinic dehydrogenase by malonate, they all are competitive inhibitions. Okay, enzyme catalysis, it is already explained. I'm not going to do it again. Just look at the picture. Non-competitive inhibition. Non-competitive inhibition, this type of inhibition is recognized by its characteristic effect on Vmax. Non-competitive inhibition occurs when the inhibitor and the substrate bind at different sites to the enzyme. The non-competitive inhibitor can bind whether with the free enzyme or to the enzyme substrated complex, thereby preventing the reaction from occurring. So non-competitive inhibitor, it can bind to the enzyme substrate complex as well as with the free enzyme. Okay, non-competitive inhibition occurs when the inhibitor and the substrate bind to different sites in the enzyme. Effect of Vmax. Non-competitive inhibition cannot be overcome by increasing the concentration of the substrate. Thus, non-competitive inhibitors decreases the Vmax of the reaction. Okay, the non-competitive inhibition, it cannot overcome by increasing the concentration of the substrate. Thus, non-competitive inhibition, it decreases the Vmax of the reaction. Effect on Km. Non-competitive inhibitors do not interfere with the binding of the substrate to enzyme. So they do not interfere with the binding of the substrate to the enzyme. Thus, the enzyme shows the same Km value in the presence or absence of the non-competitive inhibitor. So they decreases the Vmax. Non-competitive inhibitors, they decreases the Vmax, and they do not interfere with the binding of the substrate to enzyme. Okay, thus the enzyme shows the same Km in the presence or absence of the non-competitive inhibitor. So Km remains same. The Michaelis Menten constant is remains same. Is same. Vmax decreases. While in case of here, the Vmax is affected, okay, as inhibitors will decrease the rate of the reaction, okay, it will compete with the substrate, and effect of Km increases. Cyanide kills an animal by inhibiting the cytochrome oxidase. That is non-competitive inhibition. So this is the bout of non-competitive and competitive inhibition. We are left over with two or three more topics in the same chapter. Okay. Till then, we will be starting the next new topics. Please go on revising the previous topics and the questions as well. Okay, my dear students, then I'll take your leave. Bye bye. Take care. Stay safe. Stay healthy.